Get this going right on time. Welcome, everyone. Welcome to this month's Digital Cafe. My name is Christina Corral. I'm with MSU Extension, and I'm one of the PIs on the Soil Health Nexus, along with Ann Nardi, Walt Sell from Purdue, Ann's from University of Wisconsin. And then from University of Nebraska, we have Leslie Johnson, and we make up the PI for the project right now. So let me talk a little bit about Soil Health Nexus. We are a group that are university led, and we are dedicated to increasing access to research-based soil health knowledge and extension resources. We have people from all of the North Central region states, all 12 of them, who are on our Soil Health Nexus. So how we're going to handle today is we're going to have a presentation, and I'll go through our speakers in a little bit, and then we're going to have a question and answer time with that presentation. Then we're going to go into breakout rooms. You get to pick which breakout room you're going to be in. You can either go to the researchers, educators, agency, farmers, or students section. You can pick those. When we get done, you will go to those breakout rooms. Today's speaker, we have Dr. Andrea Bosch and Fernanda Krupek. Both of them are from University of Nebraska-Lincoln, and they're going to talk to us about the lessons that they learned from their soil health project. And I'm going to go ahead and stop sharing and turn it over to you. Andrea?
Great. Well, thank you, Christina, and thank you, Ann, and the organizers for inviting us to present to this great group. It's nice to see so many people here. We weren't sure how many people we'd have. It's spring break here at the University of Nebraska, so happy spring break, happy middle of March. As we were just chatting about, it may be warm or cold or all of the above from wherever you are. So what I want to talk, Fernanda and I want to share with you today is predominantly about the work she's been doing for her dissertation research, as well as the work of Elizabeth Oiss, who was an MS student who finished last year, from a project that involved all on-farm research in Nebraska. And so we're combining some soil and some weeds work that we did on several different farms across the state. And after several years, we have some interesting findings and syntheses of our work. So 3 studies to share with you, and we'll try to get through as much as we can to leave time for discussion and possibly the rooms. So Fernanda will talk about the first 2 studies, which are predominantly related to soil, and some from a larger number of farms, some from just 4 farms, and then I will share some of the work that Elizabeth did on weed communities. Predominantly, this work is focused on the impact of cover crops on farms, and then try to tie it all together, what we've learned from all of these studies. So that is where we're headed this afternoon. I did just want to remind us that, as I'm presenting this is a team effort, right? So just a quick picture of our group. This is Fernanda and Elizabeth. My dog, I like to show pictures of him, but just a reminder, he's not a part of the research team officially, but just that this is a team collaborative effort that's gone into many years of collaboration that we've had with the NRCS, who's funded this work. And so I like to share this picture in presentations that I give because it really is a motivating element of the work that I do, right? So this is a photo taken from a rest stop that is on I-80. You can access it if you are driving east. And what the middle part of these pillars here are meant to represent is the depth of topsoil, how it's changed in time since basically European settlement in the middle of the 19th century in the Midwest and Great Plains. And so essentially what it's depicting is that the depth of topsoil has moved from, at that time, about 20 inches to less than 7 inches, right? And so we know that this is a trend that is something that we all as a community are working to reverse or mitigate. And here I am at the end of this photo wondering what's going to happen in 2050, right? And so we hear a lot about, the food demand and calorie demand of a growing, hungrier planet than what we'll need to do from a crop production standpoint by 2050. But I would challenge us all to, think about this question of what will our soil resource look by 2050. So yeah, so that is a motivation behind the work. And now let me just tell you a little bit about the project, right? So this is an effort actually that got started, I'm in my 6th year at UNL. It got started in the middle of 2017, so before I came, where farmers worked with NRCS and others at UNL Extension with our great on-farm research network, which I'm sure some of you are familiar with, to put together on-farm experiments. So farmers actually have EQIP contracts as part of this project and they agreed to participate in on-farm research. And so there were 17 farms in all of these counties that committed to 5-year efforts to study different soil health management systems on their farms. And so that includes a number of different practices that people are doing experiments with, as well as what they had a site history of. And as Fernanda's going to tell you about in her first project, she put together a really great classification system, not only to categorize those experiments, but also to take into account site history. So this includes different management histories and experiments with crop rotations, some people who have livestock and using organic amendments on their farms, as well as different tillage practices, mostly no-till or reduced till, and then cover crops, right? So they are illustrating all of the different elements of soil health. And we have here for you, if you're interested, the QR code to learn more about the project, where we have all of the on-farm research reports that we published over several years. And so the reason that I go into that explanation, and as Fernanda will explain too, is that farmers were able to design the management system that they wanted to study, right? So all of them do include cover crops, but they have different experiments on them, right? So some are comparing different species of cover crops, some have cover crops and grazing, some have different crop rotations, but important to illustrate that farmers had the opportunity to design these experiments and then work with Extension and NRCS to see that these were replicated and randomized in a rigorous way that we could do more analysis on. So the requirements for folks to participate included giving us some information about their management, which is all organized and aggregated into our on-farm research reports. And then a number of the farms also had NRCS assessments taken, as well as a Haney test, which Fernanda is going to tell you about in her first study. Okay, so that's our introduction, our preamble, a little bit about the project. So all these on-farm experiments that look a little different. And I encourage you to use the chat here to just tell us your name, where you're watching the webinar from, and if you have a favorite cover crop species and why, just to give you a chance to transition into Fernanda sharing more about some of the soil results that we found from the farms. Yeah, well, we'll look for your responses in the chat. Go ahead, Fernanda.
Andrea, do you want to share with us your favorite crop
Oh,
species?
cereal rye, because it can grow and do everything. Emerges quickly, greens up in the spring. I still am impressed that it can, do as well as it does. And it is the most widely used cover crop in the US. But actually, my background here is a multi-species cover crop mix from one of the farms that is included in all of these studies from central Nebraska. So that's what my background is. And my favorite.
Awesome. Yeah, no, I agree with you. And the majority of the on-farm trials that are part of this project we are presenting here today include cereal rye, and yeah, this widely used in Nebraska and across the region. So we have some folks sharing here with us some of their cover crop species that they are favorite, oats, triticale. Yeah, thank you all for sharing with us this today. And yeah, I want to first of all, thank you for this opportunity to be here, and I appreciate everybody's time in joining us. I'm going to start here today sharing this first study that came from the farm available soil health assessments that were collected in 10 on-farm sites in 2019. So as Andrea mentioned, farmers, they selected the treatment of interest, right? The majority included cover crop in some capacity, but there is also crop rotation, livestock integration, and to add more complexity to this, differences in field histories as it relates to cash crop diversity, cover crop use. For example, we have farmers who are introducing cover crops for the first time, whereas others have been adopting cover crops for over 12 years. So there were differences there. Also in terms of mechanical disturbance, some farmers using strip tillage, others have been no-till for over 30 years. And there are also some differences in application of organic amendment and mixed livestock and cropping enterprises. So from a data analysis standpoint, you might think that there are some challenges really to account for treatment and site history effect on the soil property changes, right? So one of the strategies, let me see if one of the strategies that we adopted, we try to put each on-farm site in a scale, right? By developing those indexes that you are seeing here. So indexes in terms of cash crop diversity, frequency of mechanical disturbance, cover crop and organic amendment, and crop and livestock integration. So think about putting all these farm sites in this scale. And you are seeing here how we calculated each of those indexes. And just to give you some background, for example, time of cover crop use, like I mentioned before, varied from 0 to up to 12 years, years without soil disturbance from 0 to 30 years. Also, numbers of different plant species in a 5-year rotation in those fields vary from 2 up to 18. So the idea here is that those indexes, the greater the number of the index, the higher the number of crop diversity, fewer number of years with fall bare soil, fewer soil disturbance, higher number of organic amendment, and high crop and livestock integration. So we apply this classification approach to 21 indicators reflecting soil physical, chemical, and biological health, which you are seeing in this table here. And all those fields, they were sampled at 0 to 15 centimeters soil depth in the fall of 2019, which represents 2 to 3 years after the experimental plots were in place. Some of the properties you are seeing here, they were part of the protocol that NRCS developed in Nebraska to measure water content, infiltration, bulk density. But there are also some other properties that are part of the Haney test that some of you might be familiar with this commercial test that uses a different soil extractant than general chemical fertility tests. And there is also some nutrient recommendations that we looked at for corn and wheat. And those were developed based on calibrations from the University of Nebraska and also Kansas State University. And it's important to point out here that all those calculations, they are still evolving, right? But those we are presented here were used as of February 2020 by Ward's Laboratory. So we ran basically multiple linear regression models to try to identify the combinations of practices that perform best, right? The most important factors leading to improvements in soil properties when we consider all those indexes simultaneously, those that I mentioned before: cover crop use, crop diversity, years without soil disturbance, crop and livestock integration, and organic amendment. And the contribution of those management intensification to different soil properties was quantified with a relative importance analysis, which is this graph you are seeing here. So we observed that soil properties that are closely related to carbon and nitrogen dynamics, like you are seeing here, soil respiration, organic matter, water extract of organic carbon, nitrogen, total nitrogen, and also soil health score, they explain the majority of the total variability in our model. So cover crop use explaining the highest variability we are seeing on those properties. Oops, sorry. So in terms of crop diversity, we see here that soil water infiltration and volumetric water content explain the majority of the variability. And when we average all those soil health indicators, we were able to see that cover crop contribute to 30% of the overall influence of all those assessed variables, followed by years without soil disturbance, crop and livestock integration, organic amendment, and crop diversity. So I'm not saying here that cover crop is better than those other practices, right? But what I'm trying to say is that the future of continuous leaving cover and roots in the soil with cover crop explain most of the changes we were seeing in soil properties. And because cover crop have, let's say, the most weight on those soil properties that are closely related to organic matter and carbon nitrogen dynamics, we further explored the impact that cover crop has on those Haney test calculations of available plant nitrogen and also nitrogen recommendations for corn and wheat and nitrogen savings. So first, let's look at this first graph here. We have organic nitrogen release, which is the overall credit that Haney test calculates from the soil. The other more conventional fertility tests utilizing nitrate or ammonium do not account for. So this represents, let's say, the amount of nitrogen being released through microbial activity from organic nitrogen pools. And there are some premises for this calculation that I can go in detail if we have questions on this. But the idea here, we are seeing that intensification in cover crop use, which is represented by larger and lighter blue circles, resulted in greater 24-hour soil respiration and also organic nitrogen release. And when we look at the nitrogen recommendations here, we see that farmers using cover crops for over 8 years, this organic nitrogen release could be above 10 ppm and reduce the nitrogen fertility needs substantially. And this is what we were documenting here, that nitrogen savings for fields that were adopting cover crop for over 8 years saved on average $18 per acre in nitrogen application, compared to fields with low intensification in cover crop use saving less. So here we are being able to see that fertilizer recommendations could be reduced when we account for organic nitrogen pools as a way to capture this greater potential nutrient pool than other standard soil testing, right? So really here being able to see that soil health practices helping to increase farmers' return on investment because soil is functioning and would help to reduce the inputs. And I'm also showing here data from the baseline, which is 2016 or 17, when that program was launched, when Andrew mentioned. So we are comparing here the baseline in 2019, which was when we sampled the fields again. So we see here that when we average across all fields, the changes in infiltration and bulk density, water extractable organic nitrogen, and soil health score, they were not different from zero, right? We see that all those boxes presented here, they cross the 0% change from the baseline. But the fields we are seeing here with no history of cover crop use, we observe these trends of increased bulk density and also reduced infiltration, water extractable organic nitrogen, and soil health score compared to that initial assessment. So really, we are seeing here that reducing fallow periods with longer cover cropping showed improving trends in soil structure, water and nutrient cycling, and also overall soil health score. So just some take-home messages or summary for this first study, right? That cover crops have the most weight on soil property changes and can reduce fertilizer use when we consider those organic nitrogen pools for fertilizer recommendations. So I just want to emphasize here that investing in soil health is not a one-year return on investment, right? That process of cover crops, increasing microbial activity, hopefully turning that into biomass so that carbon and nitrogen are incorporated in organic matter really takes time. And hopefully these reductions in fertilizer inputs we are documented here are allowing farmers to be closer to that nitrogen optimization, right? Not insufficient, not applying excess, but that optimization, which is one of the most challenging aspects. It's something that requires further investigation, but we are seeing we are being able to document those reductions in fertilizer inputs. All right, so before we move to the second study, I just want to bring here some related themes that emerge out of some in-depth interviews with farmers that were part of this initiative. So we asked farmers questions related to their credible sources of soil health information, and soil health testing was a topic that they brought up. And I just want to share here some of the quotes. Some of the farmers have been using soil health testing as a way to gather information on soil health. And you're seeing here in one of the quotes, this farmer mentioning that they compare nutrient recommendations of chemical and the soil health testing. Other farmers also acknowledge that soil health testing is important, but it's still an evolving work, right? They are still waiting for more concrete evidence before making investment on this. And this quote here illustrating this farmer mentioned the need of better correlations between soil health testing and other farmer systems outcomes. And then this last quote here illustrates the nutrient recommendation aspect I mentioned before. So farmers are really eager to learn ways that they can cut in fertilizer needs with those high prices, right? So this quote here, farmer expressed the importance of tests that goes beyond chemical fertility tests and to really help them to add the right amount of fertilizer, right? Not insufficient, not in excess, but the right amount so that microbes also can work properly and do their job as well. All right, so I will move now to the second study, and the motivation behind this is that measuring soil property change with cover crop use in farmers' field requires this refinement in reference-based soil health potential and also adequate sampling intensity. We often hear from farmers, how should we sample for soil health, right? This is a common question. So for this study, we use a subset of 4 fields that were part of this initiative. And those fields, they were integrating cover crops for the first time. So all of them had a cover crop and no cover crop strip trials. And we also included in the soil sampling protocol reference sites. And those were grassland sites adjacent to the farm field and this approach has helped us to add some value on how we can learn more and detect differences with cover crop adoption, especially in the short term. We are talking here 3 years after cover crop was introduced. So the concept of reference stage has been around as an ecological concept, and the idea is that the reference stage reflects areas that have been less affected by human activity, either a native virgin land or undisturbed soils with minimal degree of anthropogenic modification of soil properties due to management or land use change. So we are using here grassland areas for soil health benchmark because those areas embody the 4 principles of soil health, right? And we use this reference state to compare agricultural management and to understand what is the potential, right, of that agricultural soil with different management, in this case, cover crop and no cover crop, to achieve some outcomes or improvements. So what we have done here, we are calculating for different soil properties, the relative soil health. So if you think about the reference state being 100%, right, which is in the denominator of this equation here, then the agricultural managed area, we have cover crop and no cover crop treatment, and we are interested in how close are those various soil properties to the reference state. And then the second point, we are examining the effect of sampling intensity on the power of water infiltration measurements to discriminate reference sites from those undergoing soil health management with and without cover crops. So essentially, we are taking repeated water infiltration measurements, and we are seeing here how we did that. We collect up to 5 repeated measurements, and they were subsequently averaged for each sampling point. All right, so I'm showing here in this first graph the 4 farms, all of them located in central and east central Nebraska, and the relative soil health score for 6 different soil properties. Those properties you were seeing here at the bottom. So those properties usually follow a higher the better pattern, right? Which means that higher value would, in general, signaling improved soil function. So you are seeing here bars on the left are the cover crop, on the right are the no cover crop, and the dashed horizontal line essentially represents 100% score of the reference site. So 100% of all those 6 soil properties. And we can see here that for all the sites, the aggregated bar is higher for the cover crop treatment, meaning that it has a total score that is trending closer to the reference state. And you can also see here the stars, which basically means if there is a statistically higher value for a specific property at a specific site. So we see that at least one property improved at every site. For some sites, there was 4 properties out of the 6 analyzed, but overall, cover crop is trending closer to the reference site. And the same apply for some properties that follow a lower the better pattern as well. Okay, we also look at corn and soybean yield and in each of those on-farm strips. We generally see a weak association and that has been documented in other studies as well, but there is this trend that the higher the relative score or closer that the soil on-farm site is to the reference site, the greater the yield. So the circles here representing cover crop treatments were higher at most of the sites. So I think this is very interesting, although with the weak association, we're still able to see this correlation between soil change and crop productivity. All right, and I want to show you also in terms of sampling intensity. I know this is a very complex stats method that we use it here to try to separate those treatment differences. So please do not worry about all those vertical, horizontal axes you are seeing here, but just focus on the different colors and shapes that are the treatments here. So the reference sites are represented in a square, cover crops are circles, and no cover crop is a triangle. So we can see that when we only look at 2 samples per sampling location, all those treatment symbols, the colors representing them, they are all together, right? But as we count from 3 to 4 and then 5, we see that those shapes are discriminated or differentiated from each other. So really meaning here that the greater the sampling intensity, the higher the likelihood of properly accounting for variability in field measurements conducted at the farm scale. And this is here how we can tell that there is a difference between cover crops and the no cover crops. So in general, what are some of the take-home messages here? We were able to see that cover crops close the soil health gap, but on-farm experiments must be sampled intentionally, right? We observed that selection of reference sites that embody soil health principles are important, especially when we have soils lacking example of long-term soil health management adoption. Also, in terms of number of samples, I know that there is a trade-off between number of sample and resources, right? Cost and time. But we were able to see that up to 5 random measurements per sampling per soil type, sampling every 4 to 5 acres resulted in greater separation of the treatments. And this is my last slide here. I want to close this study with another related theme that emerged out of the in-depth interviews with the farmers part of this initiative. So we asked farmers about their perception or their cost and benefit analysis of different practices intended to improve soil health. And 2 themes were predominant in these conversations. First, that closing the gap in soil health is context specific, and the second, the importance of long-term on-farm experiments to account for climate and site-specific variability. So I'm showing here this first quote illustrating farmers' observation that cover crops has worked well on some of their low-yielding environments and made yields less variable. So here, thinking about the role that soil management has in washing out land use legacies. And also, the second quote here, a farm bring the site specificity aspect that I mentioned with, there are many options within the cover crop spectrum to customize for each farm and ranch scenario, right? And this last quote here on the importance of long-term on-farm research. So this farmer here mentioned the importance of multi-year studies, thinking on 3, 4, 5 years. Those are the studies that farmers gain the most out of that research information, right? Because we are able to document how a practice performed in a challenging year, in an average year, in above average year. So those are some of the themes that emerge out of this conversation that I just want to bring here as it relates to the data that we also presented. So I will hand it over to Andrew, who will continue the presentation.
Great. Thank you, Fernanda. So we've just heard these two great studies. I dropped those links in the chat for everyone that Fernanda also had in the QR codes. And want to shift from some of the learnings we've had on the soil side to some things about weeds. So this is the work that I'm presenting that was predominantly done by Elizabeth Oyce, who was a master's student that finished here last year. And so we were really interested to understand if there'd been an impact of cover crops on the weed communities at these farms. And so why is this an interesting question, in particular in a place like Nebraska? So Nebraska has a lot of cropland. We're number 1, 2, and 3 in a number of major commodities, including cattle and corn. And we have a lot of no-till acres, right? So we are especially where we are here in Lincoln, the western edge of the Corn Belt, we have one of the highest percentages in the Corn Belt of no-till acres. So lots of no-till and lots of corn and soybean. And that is important to note because as you all are probably very familiar with over the last several decades, I like to remind my undergraduate students who are mostly born in this century that they have grown up only in an era or predominantly in an era of chemically based weed control, right? And so as we reduce tillage, a positive thing for soil health, the trade-off or one of the trade-offs is, along with the reduced diversity in cropping systems, that we are relying on chemically based weed control, right? And so what are other cultural practices that we can help producers with respect to cultural practices and just integrating weed management, right? So we know that herbicide resistance has grown more common in places like Nebraska, across the Corn Belt. We have a number of, in the double digits of herbicide resistant weeds and many cases of multiple herbicide resistances. And appreciate, this note here that herbicides have been available for less than 100 years, and we've already seen evolution in some of these challenges. And this point that the companies have not created new modes of action for several decades, right? This is something actually just right before spring break I talked to my students about, and we just did a little digging as to why no new modes of action have been developed, right? That you might already be very well aware of the, decades and, high expense it takes to bring products to market. And that many of the modes of action and low-hanging fruit in terms of those pathways to kill weeds chemically have already been exploited, right? So it's getting harder and it's, very expensive to do this, right? So what are the ways that we can help producers with integrated weed management? Cover crops are increasingly being looked at for that. So that was a lot of the motivation behind this work. How well will the products continue to work for us in the future depends on how well we can integrate other approaches into weed control. So in this project, Elizabeth looked at two elements of weeds, right? The first was, what is the community of weeds lurking in the soil seed bank, right? So for this study back in 2021, we had a really big effort. That's what this photo behind me was taken from 2 springs ago. Harvested soil from 4 on-farm sites that I'll show you, the 4 that Fernanda just talked about in the last project. And the reason we did that was because they were the most similar in having a standardized control with no cover crop and a cover crop for several years. And so we looked at the weeds in the soil seed bank, and we also looked at what emerged during the growing season at 2 points, as I will show you next. So here are the locations of the 4 on-farm studies. They turned out to be kind of in east and central Nebraska. So those 4 counties in red that you're seeing there. And then we worked with 2 longer term research sites in eastern and central Nebraska, in part because they had cover crops grown for 4 to 7 years. So not extremely long term. You might think about them as medium term, which I actually, we came to realize that could have an important impact on our results. So here's the 6 sites where I'm going to show you the results from. And again, 2 springs ago, see if I can get this to play, we went and just pulled some basic soil cores and aggregated them on the experiments and brought them back to the greenhouse. And what Elizabeth did was to count and categorize over 6,000 weeds that emerged from all of the trays. I'll show you some neat photos there. So we'll show you estimates of the weeds per meter squared. So we have the diversity of weeds, and we also have the amount of weeds that emerged. And then at 2 points during the growing season, right after cash crop emergence, before any post-emergent herbicides were applied, was one interval where she looked at above ground weeds, and then after those post-emergence herbicide applications about 3 to 4 weeks later. So really looking at some of these critical weed periods in that first measurement, and in that second measurement, if the cover crops were having an impact later on into the growing seasons. There's some good evidence in the literature that that first time point is when a winter cover crop in a corn soybean rotation can have really good effect. But what effect are they having later in the season as well? So this is kind of what it looked like. There's Elizabeth smiling. The greenhouse is a really nice place to be in the winter time and when it's colder. And so this is what the trays look like, right? So again, brought those soils back to the greenhouse, watered them twice a day for 7 months. After all the weeds emerged and nothing new came up for a few days, she would re-sieve the soil and kind of start the process over again. And so she did that 3 times until basically no more weeds were emerging from all those trays. And so I'm just going to start with the results of the overall weed seed bank, right? So again, this is just what was in the seed bank. This is one way to measure what's in the seed bank. It's not the only way. There's no easy way to do this. That's why I think a lot of studies don't do it. But what was interesting, again, 6,561 weeds were categorized from 57 different species. And you can see that from all of these locations, when you counted them together, that Palmer amaranth was the #1 most dominant weed that we saw, but that other amaranth family weeds were pretty dominant as well. So you can see there red root pigweed and common water hemp making up good percentages of the total as well. So that is important just as we think about herbicide resistance and the trouble that is, especially in I just got an alert that my Zoom might close in 4 minutes, but we'll hope that doesn't happen on my computer. So as we think about herbicide resistance and the issues that farmers in the South have had with Palmer amaranth, we know that this is important, right? And so here is a really interesting finding that we did not expect to have, which was that we found at 3 of these 6 sites, so I'm showing you these, all 6 sites here, that at 3 of the 6 sites, pigweed actually increased in the cover crop treatment. So imagine these are sites that have a strip, replicated strips of cover and no cover, that statistically significant differences in pigweeds at 3 of those sites. So 1 of the research sites and 2 of the farms. And in some places by a very significant amount, 180%, 243%, 355% at Merritt County. Merritt County actually still uses strip till, so they have the cover crop strips, but also uses strip till. So there was all the other sites were no till, some longer than others, which is also important in some of our findings that I will summarize for you soon. So pigweed increased in the soil seed bank at several sites. But what was interesting on the other side of that is that when Elizabeth went back during the growing season and measured when or what actually was coming up, so you can see those 2 time periods, early and late, we did not see that there was an increase in what was emerging. In fact, we found that there were significantly less weeds, not necessarily less pigweed. In some sites and some intervals, we did find that. But that weeds were reduced significantly at in the cover crop treatments at some of the sites. So that wasn't, again, necessarily just about pigweed. It was in some places more related to grasses. But what we saw in the soil seed bank was not what we were seeing. Exactly the same as what we were seeing is what emerged. So yeah, these reductions here, at least at Greeley County, by about 90%. So pigweed density, again, in spite of those differences in the soil seed bank, we didn't see that being expressed above ground. But I want to also shift and think about the diversity of the weeds, which is a really interesting element in as an indicator of what might be happening in these environments, right? So what you're seeing here is another multivariate statistical analysis where we tried to look at what was the difference in the communities of weeds at all of these sites. And what we found, and where you see these polygon shapes separate, it means that there are different, completely different communities of weeds at the different locations, right? And so here in Howard County, there was pretty similar communities of weeds, but at some of the other locations here, I'm showing you Greeley and Merritt County, where we did see that increase in pigweed, there was complete separation of the communities of weeds in the seed bank, right? So really interesting, as I'm going to get to in just a minute. And so what we also found is that even though there was not a statistical difference in the diversity, so Elizabeth did a number, as you're seeing here, of calculations for seed bank diversity, we found that the diversity actually increased. So this change here is the check minus the cover crop score. So where you see these negative values, that means that there was an increase in the diversity of weeds in those cover crop strips. And why we think that this is so interesting is because from an ecological standpoint, we understand that more diverse weed communities can be an indicator of a potentially healthier environment where none of those weeds are out competing each other. So, and I've seen some other research pointing to this too. Again, when I say that these sites were in cover crops 4 to 7 years, what it could mean is that as we might monitor, and which we're not necessarily intending to do, some of these sites for a longer period of time, we might even see more change, more diversity of the weed communities at these sites. Okay, so just to the speed through of these 2 years of Elizabeth's work, again, we saw that the cover crops were suppressing weeds, including pigweed seeds that were in the seed bank during the growing season when we did these measurements in 2021, even though at several of the sites, the cover crops were increasing weeds. And so we do think that this suggests to us that cover crops could be a great tool as part of an integrated weed management plan. And, you know, this is kind of an interesting finding, and maybe people may be really curious about some of this. When I share this with some of my students, they had some questions to my undergraduate students. Again, this is fresh in my mind because I just shared it with them right before.
I think Andrea had some.
Yeah, I think it looks like we might have lost Andrea.
Let me.
Oh,
Well,
wait, maybe here she's
I'm
back.
okay, good.
Well, here's a lesson in why I've been away from my desk for spring break and not updating my computer. So update your Zoom when it tells you to and don't delay it. That's what happened. So sorry for that. So again, the benefit that cover crops might have in an integrated weed management system, of course, this adds complexity. I'm sure that many of you who are, from the names that I recognize, experienced conservation professionals know that cover crops add a management complexity, but that they can provide benefit in the longer term as well. Fernanda, you may need to advance slides for me now.
I think it should be.
Am I?
Yeah, go ahead. I can.
Okay. Okay, and again, another QR code for our project. So cropwatch.unl.edu/soil health. You can see links to all of those on-farm research reports in the sites where we had more soil health data and some of the weed management data. We have included that on those on-farm research reports as well. I did want to make note too, for those of you who are with NRCS or who interact with farmers, the reference site sampling can now be included in a conservation practice code. And so that's one of the things that we think is really worthwhile from some of Fernanda's work, just this adding more evidence to the idea that reference site sampling can add to our understanding of how our management is impacting our soil. So just wanted to make note of that if you're not aware that that's conservation code can include reference site sampling as well. And so thank you again for your great attention here this afternoon. Sorry for some of the technical issues that we had. And again, thanking Elizabeth and Fernanda for the great work that they've done here as well.
Thank you. So there are some questions, a couple of questions in the chat. If Andrea, if you and Fernanda want to answer those, I can read them if you'd like.
Sure, I can read
Okay.
Colton's question, right? So you said these sites have been using cover crops for 4 to 7 years with some programs lasting 3 years. What happens to the pigweed seed bank if the cover crops aren't planted when those programs are over? I think that's a great question. In fact, this is similar to what some of the students brought up with us as well when I talked about this in class. I mean, it's possible that without that competition from the cover crop that you could see those weeds emerge. It's hard to say, but you know, if just like if you stopped using an herbicide or another element of weed control, that those weeds might reemerge. I don't know, Fernanda, you want to say anything that you talked about or learned about with your farmers in the interviews with respect to continuing on with cover crops after programs are over?
Yeah, absolutely. That was actually part of the motivation to do those in-depth interviews and some of the quotes that I brought here, but the majority of them, they were able to see the benefits of cover crops over that 5-year program, and they were interested in continuing that, for sure, with some adaptations, right? Depending on if we are expecting a more of a drier year, they might do some tweaks on the species selection, but they were interested in continuing those studies. And I see a question here. Study 2, was the soil texture the same between the reference site and no cover crop site? If not the same, how did you account for inherent soil properties in your analysis? So, yeah, that's a great question. And yes, the texture was the same, and that's why we were trying to be conscious on what points we were collecting soil samples on farms. So we try to identify, like I showed you before, there was that randomized strips, but we tried to select soils within that on-farm site. There were the same soil types representing up to 50% of the entire area, and they were similar to the soil texture of the reference site.
Right, there's a couple more questions in the chat box.
I'm trying to find them here.
I'm only seeing since I rejoined.
Christine, would you mind reading some? I'm just trying to.
I'm just making sure we didn't, I'm gonna read them, and if we've answered them, you go up at the top. Scott asks, how would you collect fields for evaluating the increase of hydrologic function, reducing runoff detention, flood reduction?
That's a great question. In fact, when I started this program back in 2019 was when we have the flooding event in Nebraska, and some of the farmers, what was interesting, they were able to see some of the differences in water infiltration in their fields compared to a neighbor fields or in the area. So great question. How would you, so can you repeat again? Just how would you?
how would you collect fields for evaluating the increase of hydrologic functions such as reducing runoff, water detention, flood reduction?
I think water infiltration, saturated hydraulic conductivity is one of the measurements that has been used and adopted to understand better the potential of, let's say, flooding mitigation. I don't know, Andrew, if you have any thoughts on any other specific properties?
Yeah, I know Scott is very keen on this question that I'm very interested in as well, which I think what you're getting at could be related to like, how would we assess this maybe at a larger scale? I mean, I think the management and site history is really important to know. I think if you have, and I've seen some studies that attempt to do this, a watershed, let's say with more conservation than another that is nearby that has less. That would be another way to try to answer this question. So knowing some about histories and management over time. I know that on a larger scale, I know there's some work with folks at Kansas State and NRCS funding where they've looked at field scale, like larger replicated experiments where they have like kind of runoff collecting flumes. I've not been involved with any of those in the past. I know that they're challenging to set up and that's why people don't do them as much. So I think that there's ways, you know, what Fernanda did in this study, we had those strips with and without the cover crop, measuring a few properties related to water. That's one way it could be looked at, but I think at a larger field or watershed scale, there's some other ways we could make approximations of this. I think the history is one of the most important things coming to like selection and drawing bounds around what we might want to study with a question like this.
Right. So another question in here is, Andrea, you said that these sites have been using cover crops for 4 to 7 years with some NRCS cover crop programs lasting 3 years. I'm curious as to what happens to that increased pigweed seed bank if cover crops aren't planted after those programs are over with. Right.
Yeah. So Colton's question, like I said, I think that they could reemerge if we reduce a weed management approach. I'm seeing some of the newer questions now too. I see Rob's question, and hi Rob, I'm glad you joined. Any ideas why the weeds were greater with the cover crop? So we do have a few ideas. We think that this could be that if the cover crops are suppressing emergence, that could mean that, you know, if you think about a seed in the soil, and if it emerges, then it's no longer a seed, right? It becomes a plant. So if there were less plants emerging, it could be that there were more in the soil seed bank. And I also think that it could be a part of this succession of the community that if we continue to monitor this, would we just see more pigweed? That was where we saw the biggest differences. And I see this other question about, was there a count of the known herbicide resistant weeds to see how cover crops are suppressing it? So that's a great question. You know, we only categorized the weeds were small when they were measured in the field. So Elizabeth separated them by grass and broadleaf. And so we did see reductions at some sites in either grasses or broadleaves. I would say my understanding is that many of the herbicide resistant weeds are broadleaf. So I don't know that we could necessarily answer that question exactly based on, you know, unless we let those weeds get much bigger, which we did not in the field.
And just to add, those 4 sites that Elizabeth did her sampling was those farmers, they were implementing cover crops for the first time. And that's why they had a no cover crop strips. Those 4 farms, or the majority or the rest of those involved in this initiative, they didn't want to have a no cover crop strip because they were already more advanced in cover crop adoption and were interested in learning other things with the species selection. So those 4, with and without cover crops, they were first time introducing cover crops in those fields.
If we've missed your question, can you go ahead and put it in the chat box? But we're doing that. I'm gonna share my screen real quick and well, you can probably see more than you want to see there. So next month, we're gonna keep on this whole cover crop topic, and Dr. Rob Myers is gonna come back. He was with us last year and he did a phenomenal job, and we got requests to bring him back. So he's gonna come back and he's gonna talk about the top 10 impacts of cover crops on soil health. And we would love to have you come back, tell your friends, and just have them come join us. So if there's no more questions, we have been on for an hour. Thank you, Andrea and Fernanda. You did a great job. We're going to forego the breakout rooms this month, if everybody's okay with that. And we just want to thank you for coming. And we'd like you to come back next week. Or next month. Don't come back next week, next month. And again, if you have any suggestions on future speakers, please let Ann Nardi or myself know, and we will do what we can to get them to come and talk to us. So thank you for joining us this month.
Thanks, everyone.
Thank you.
Olivia, yes, if you want to go to the Nexus website, we do have a Zoom link. You can register for next month's program.